Welcome back to botany class, students. We are discussing about the NEET points in the cell division chapter. That is in the first year cell division chapter. Okay. So what is the cell division and how many questions we will get? We will get nearly 2 questions from this chapter. Those are the direct questions, and 1 question will be coming in the indirect manner. That is along with the families and the microspores and the megaspore formation. Along with that, one word problem, that is the guessing only. We can get, we may not get. Okay, and usually we will get 2 questions from this. So what could be the most important concepts in this and how to prepare at the first? So during the cell division, the cell has to divide and make the new cells to be adjusted in the same place. So for this condition, we will be having the 2 types of cell divisions, and these 2 types of cell divisions occur in different types of the cells in the organism body. So one will be in the somatic cell, that is in the organelle formation cells, and another one will be the reproductive cells. In these 2 cells, 2 types of cell divisions will occur. In the reproductive cells, only the meiosis and the mitosis can be seen. Meiosis is must in the reproductive cells, whereas in the somatic cells, only mitosis will occur, which will be considered as the simple division, where one cell is going to be divided into 2 daughter cells, 2 equal sized daughter cells. That is the mitosis. Whereas in the meiosis, one parental cell undergoes meiotic division to form 4 haploid cells. That is, the chromosomal number will be reduced over here, whereas the chromosomal number never be reduced in the mitosis. And what could be the stages in this? So just I am sharing the screen, just have a look at this. See here, in the mitosis, it occurs in 2 steps. That is the cytokinesis is the last step, and the first one will be the interphase and the M phase and the cytokinesis. In the M phase, cytokinesis will be there. So here, interphase means the resting period between the 2 cell divisions. Otherwise, the starting of the cell division is the interphase. What happens in the interphase? The cell will prepare itself to go with the cell division. That is, that may be the mitosis or the meiosis, anything. But this interphase is must in every cell division. Okay, and after that, this will be in the interphase sometimes, not in sometimes. In the mitosis especially, we can say the S phase, the G1 phase, S phase, and the G2 phase will be there in the interphase. G1 phase means where the cell will be enlarged and much more food is going to be stored in it and prepared in it, and the RNA genetic material is going to be formed in that. And the S phase is the synthesis phase. What they will ask, what is meant by S phase in the cell divisions? So S will be, don't get confused with the ribosomal S subunit and this. So there they will say measuring unit. S is called as. Here S is simple S phase, PHAGE. Okay, S phase means that is nothing but the synthesis phase. What happens in the synthesis? DNA material is going to be replicated, otherwise the duplicated. Okay, that means if the 2C chromosomal 2C the genetic material is there, then that becomes a 4C. But the basic chromosomal number that is there for each and every organism never changes in it. That is the S phase. And the G2 phase is the last phase, where gap 2 phase also we can say, where the cell will be enlarging much more and much more food material is prepared and stored in it. That is the G2 phase. Now this will be going into the jumping into the mitosis phase. That is the M phase. So in this one, it is having 4 steps: prophase, metaphase, anaphase, telophase. In these 4 steps, what happens? In the prophase, the chromatin material condenses to form the chromosomes. That is, it making the chromosomes according to the organism body. So how many number of chromosomes has to be there? It will make that chromosomes only. And then in the metaphase, So the equatorial plate is going to be formed in the center of the cell, which will divide the cell into two cells. Okay, that is the equatorial plate, and the spindle fiber formation from the poles, and these spindle fibers will attaches to the centromere of the chromosome and makes the chromosome to be placed on the equatorial plate. Then in the anaphase, the chromosomes will be splitting into the centromere and pulling to the respective poles. So in this, the centromere will be dividing and the chromatids will move to the respective poles. Like that, same number of chromosomes will be present in both the daughter cells after the telophase. So what they will ask over here, so in which division the spindle fibers formation starts? That is by the end of the prophase, otherwise by the starting of the metaphase, what we can say. Equatorial plate formed in the metaphase. And when the chromosomes will move to the respective poles, at which stage? That is the anaphase. And at which stage the nuclear membrane, nucleolus, and the all other cell organelles will reoccur. That is the telophase. In which phase all these ones will disintegrate? That is in the prophase. Like that only we will have the questions. And after the telophase, cytokinesis means the cell divides and makes the two equal sized daughter cells. That is the mitosis. And now comes the meiosis. Meiosis means which is the reduction division. And in this reduction division, the cell divides into 4 haploid daughter cells, which are unequal. All 4 are unequal. Okay, so 50% are similar, 50% are dissimilar to the parental cell. What we have to say, not in between them. These are dissimilar to the parental cell. That is the meiosis. In the meiosis, again, 2 steps will be the meiosis 1 and the meiosis 2. Meiosis 1 and the meiosis 2. And the meiosis 1 is the original reduction division where the chromosomal number is reduced to half, and this is the lengthiest phase also. In this, again, prophase is the lengthiest phase in the meiosis 1, where the 5 substages are there. All these 5 substages, they will ask to arrange them in the order, in the master following. Otherwise, in this, which substage is the lengthiest one? Otherwise, in which substage the chromatin material is going to be dividing? Otherwise, overlapping of the chromatids will occur. All these things they will ask in this. So that is the packaging is the biggest subphase in the prophase 1 of meiosis 1, where in this one only the overlapping of the chromatids, the chromosomal arms will occur. That is the most important one. So these 5 substages are leptotene, zygote, packaging, diplotin, diakinesis. In this one, packaging is the lengthiest one. Okay, then the second lengthiest subphase is the diplotin, and the diakinesis is the precursor of the mitosis. And in which type of stage the complete reduction of the chromosomal number occurs? That is the anaphase 1, where the chromosomal number becomes half during the cell division. So the original reduction division occurs only in the meiosis 1, and the meiosis 2 is the just simple, otherwise similar to the mitotic cell division. What they will ask this one only. Okay, and prepare well from this, and the diagrammatic questions also may come. It cannot be said, but usually they won't give. In the EAMCET exam also, they will ask these 5 substages and the characteristic features of these 5 substages. It's very, very important. And the prophase 1. And all these are belonging to the prophase 1. They will ask like this and prepare well. Okay, right. Thank you, both.